Hello everyone, my name is Ashfaq Ahmed and I work in the field of bioinformatics and structural biology. Welcome to Bioinformatics Insights. Today we will talk about how to perform QSAR analysis. I have divided these learning modules in two steps. It means the QSAR analysis will be covered in two videos. Today is video 1 and the next video will be video 2. In video 1, we will work on the data generation and we will understand the basic concept of QSAR analysis. In video 2, we will use the data generated here for QSAR model generation or model construction. We will train and test our model, we will optimize our model, and finally, we will make some predictions. So let's start part 1, which is data generation for QSAR modeling and understanding of QSAR models. So first of all, you have to open your browser and type colab.research.google.com /GitHub/ash100. I will also drop this link in the description section of this video. So you need to click and then you will see this kind of window. Click here and then click ash100 /CAD_project. Then scroll down and you will see part 14, QSAR modeling, descriptor generations, part 14. Click here. Okay, now connect. Okay, what actually is QSAR and why we perform it, how much important it is, and how we will prepare the data, we will discuss all in this section, all in this video. Okay, QSAR stands for Quantitative Structure Activity Relationship. It means that you are interested to probe the relationship between a compound activity and structure. So every compound has a different activity in the form of IC50 or KI or PIC50 or whatever, and these compounds have distinct or different structures. So the QSAR analysis are basically aimed to elucidate, to validate, to identify or to highlight the relationship between the structural parts and the activity, or I would say the functional parts of the compounds. So in this section, we will use RDKit and I will educate you along the way. So first of all, you need to click here. It will install RDKit and it will also install some other softwares. Okay. It will take almost 10 seconds. Now you can clear it or you can leave it as it is. We click here and it will import the necessary libraries. Okay. So the first thing is that what kind of data we need for QSAR analysis. The simplest answer is we need a data of compounds, organic compounds, and the data should contain two things or must contain two things. One, smileys, to PIC50 values. You all know, because this is almost part 14 of the CATS course, you all know that smileys are the linear strings or the linear representation of the molecular structure. And it contains variety of information in the form of fingerprints, in the form of molecular features, in the form of Lipinski descriptor, so on. And PIC50 is a kind of activity value. The higher PIC50 means a good activity or a better activity. If it is inhibitor, the higher PIC50 value means a higher or a better inhibition compared to the one with a lower PIC50 value. So, our data should contain these two columns, smileys and PIC50 values. And we will load our data here. Why we load our data here, we will discuss below. Okay, I will use the data, just randomly some data I should have in my computer. Let's see, in my download section. Okay, wait, wait. I need to use descriptor QED, 3D descriptor, Lipinski descriptor, model performance, predicted activities, much descriptors. Wait. Okay. Okay. I don't have right now. I will use this data. So what I need to do, I will remove all these columns. Now we have two columns, smileys and activity. If you have your data, then you will see PIC50 here. So change that name to the activity and change the canonical smileys to the smileys like this. So, okay, now you may have a question that, Professor, you had this data and you deleted some columns. From where I should get my data? A good question. So, if you want to have your data, you have to watch exploratory data analysis, and there you are going to generate your data and save your data. And then we perform some exploratory data analysis in part 13. So you can use that data before starting your exploratory data analysis or those figure and charts. You already saved almost 1, 2, 3, 4, 5 data sets. So you can use any of them. Also, we have part 1 where we have used the Kimball database and extracted some data of this kind. So if you have that data, then if you want to test these things, so just I would suggest you will see canonical smiles in your data, convert it into smileys and convert the PIC50 into activity like this. So you will have a data of this kind. Okay, now let's say, okay, no missing values. Okay, I will save this data. Let's say save as, and I save it for QSAR, and I will that is fine. Okay, browse. Okay, I will save it in the download section and I will name it QSAR. Okay, that is fine. QSAR. Save. Okay, the data is saved here. Now cancel. Okay, we have this data, QSAR. We will use this data. Now, what we should do, click here and click here. It means we need to upload this data first to the notebook. So click the download QSAR. Okay. Okay. Now we have this data. Now click these three vertical dots, copy path, and paste your data here. But try not to disturb these two. I don't know how to say. Okay. And click run this. So now our data is uploaded to the notebook. Okay, now we I told you from where to obtain the data. Still, if you did not understand what I mean, then click here and watch this EDA tutorial. You will have your data. Simple. Okay. Once you have your data and you upload your data here, so stop it here. We will discuss a 2 or 3 minute theory. Okay, there are something we call it descriptors, or in video 2 or 3 in this course, we call it features. If you remember Matthias and those fingerprints. Okay, here we call it descriptors. For a molecular structure or for a molecular compound, there are different descriptors. In this video, I will teach you or I will show you how you can calculate them. We will calculate 4 different kinds of descriptors. To the one, we will call it topological descriptors. To the other, we will call it Lipinski descriptors. And to the third, we will call it 3D descriptors. And to the fourth, we will call it the QED, or the quantitative estimation of drug likeness score or descriptor. And then finally, we will merge all these 4 descriptor into a single file, and that single file will be used for QSAR modeling and for testing and training and for the prediction of unknown compounds. And I will also tell you how you can use these things in your research, but that will be part of the second video. So here we are going to calculate descriptors for our data set. First, we are going to calculate topological descriptors. Topological descriptor means topology is an English term, and it means the overall structure. I should say the overall physical structure and the architecture. So it means all the descriptors we are calculating here are belongs to the structure of the compound. So the first one is Wiener index, the second one is Zagreb index and TPSA, Balban index and kappa 1, 2 and 3. You can read these simple explanations and you will understand what that means. For detailed understanding, you can read different research papers. Okay, so in these notebooks, I have hide hidden. I have make hide the codes. So if you want to see these codes, no matter, you just you need to click here. Like let's say, see show code. So you can see this is a code for the calculation of topological descriptors. Okay. Now, this was the introduction for the topological descriptors. Now we will click here, and if you see at the end of the code, we will have a file. Its name will be topological descriptors.csv. If you want to change it, you can, otherwise leave as it is and run this. Once you run it, you will see a new file here, and you will see this kind of instruction: descriptors calculated and saved. Where? here and topological descriptors.csv. So you can click refresh, you can see here one file is generated. Now we are going to calculate Lipinski descriptors. In Lipinski descriptors, we will calculate six things. Four are the Lipinski, which are also included in the Lipinski rules: hydrogen acceptor, donor, log P, molecular weight. But two extra things I have included: number of rotatable bonds and number of rings in a structure. So just click here, it will calculate the Lipinski descriptor and it will also generate the result, and result name will be Lipinski descriptors.csv. You will see the file here or refresh it. Okay, it's here. Now, the third thing or the third descriptor we will calculate. So this descriptor will take some time because it's a 3D descriptor. It means it belongs to the 3D shape, and because of that, uh, the program will first generate convert the linear representation from the smileys to the 3D, and then will calculate those descriptors. So it will take, I have almost 2000 compounds or 2100 compounds in this data set. It takes 3 minutes. So it depends on your data set. So you can expect a time likewise. Okay, I have run the calculation and now I'm going to pause the session. So so that not to waste your further time. Okay, welcome back. It's done, and we should have a file 3D descriptors.csv here. If you are not seeing here, right click, refresh, and it's here. Okay, now, and the fourth thing we will calculate is QED, or quantitative estimation of drug likeness, or in simple term, we call it drug likeness score. Click here, and it should not take longer. Okay, QED score. So now you can see 5 files here. 4 for descriptors we calculated, and 1 is our input file QSAR.csv, topological descriptor, Lipinski, 3D, and QED descriptors. I will download, or you will need to download it for some manual processing. Download this one, also download this one, then download this one and this one. Okay, what kind of manual processing we will perform? So you need to understand, we will perform two things. In the next section, we are going to merge all these 4 files into a single file. So we will use a function, and in that function, we will use a common column in all these 4 and will merge the data. I have choose to use the common columns of smileys. But if you remember, in our data, we have 2 columns, activity and smileys. So it means in every data, we have 2 types of columns for smileys and activity. So in order to avoid any kind of error, we will rename the activity column in 3 datas, or you can easily delete it. That's not a problem. Okay. Now, the second reason is for manual editing, that sometime for some descriptors, the values are too high and the program makes it INF. INF means infinity. So for such values or for such compounds, they will make some troubles in the next section. So therefore, we will quickly remove those values or those compounds manually. So let's see. you can follow me. Uh, all the files are in my download directory, and these are the four files just downloaded at this time. So I will open it one by one, and let's say open it. Okay, 3D descriptor, don't convert. Okay, now I have, you can see I have uh, molecular volume, surface area, radius of gyrations, smileys, and activity. So I will just write in activity underscore one, and just quickly check, can you see any missing column or missing stuff? Okay, don't check it right now. Okay, just save it, close. Now open this file, topological descriptor, don't convert, and make this activity 2 underscore 2 and save it. Okay, now open Lipinski descriptors, don't convert, and make this activity 3. Okay, and save it. Okay, now, but I did not convert the activity of the activity column of the QED score. That's okay. We have changed the names of the activity column for 3 data sets and will remain the one as it is. It's up to you which 3 you need to change. That's not hard and fast rules. Okay. Now what to do next? Come back here. You have these 4 files. You can see here. So you have two options, either to delete these 4 files or upload the new 4 files. I would say, okay, we did not bring any change in this one, so it will stay here. We made changes in these 3 files, so it's better to delete it. Delete file. Okay, delete file. Okay, and delete file. Okay. I hope I'm not making you confused. This is the input data. This is the QED scores descriptors we calculated, but we did not make any change offline. So I left it here and click here, and I will upload these 3 files, not the QED files, because the QED is already here. And you can see it's uploading. Okay. Now, what is next? Click here, and the QED file, you see the QED scores. Its name matches here, so leave it as it. It is 3D file. So this is the 3D file. Click here, copy path, and paste it here. Control V. Lipinski. Click here, copy path, and Control V. And descriptor file, which is for topological descriptor. Here, copy path, and here. Okay, what to do? Click run. Now it says CSV files merged and saved to content. This is the content directory, and the file name will be merge descriptors.csv. You cannot see it here, just click refresh here. Now download this file. Merge descriptors.csv. So we will use this file. Okay, let me show you. Uh, refresh. Okay, mud descriptors. Now open this file. This is the mud descriptor. You can see activity 1, activity 2, activity 3, and activity 1. We need to delete these activity columns and we need to keep only one activity column. So I think this one we will keep it because all the data in these activities are same. You can see 16.48, 16. So click here, right click, delete. Click here, right click, delete. Click here, or I would suggest do not delete this one, delete this one because we need the activity columns at the end. It will be better and change it to activity. Okay. Now see this file. These are the smileys, then the QED, then the molecular volume, then surface area, radius of gyration, molecular weight, log P, hydrogen donor, acceptor, number of rotatable bonds, number of rings, and Wiener index, and Zagreb index, TPSA, and Balban index, kappa 1 to kappa 3, and activity. So in next step, we will use all these features for every molecule and we will generate a model against this column activity. And these all are the descriptors. Remember, this method which I am teaching you, it is not limited only to these descriptors. If you want to include more descriptors, then you can calculate and likewise you can add you can merge 5 files into one or 6 files into one, whatever. Also, the other way around, you can go with some specific descriptors. It is not compulsory to calculate all the descriptors I'm teaching you. You can go for 3 descriptor, 4 descriptor, 5 features, I would say, not descriptor feature. So each of it is called feature. So you can go for some of them. It depends, and these are optional. And I hope I will not receive a questions in comments that how many descriptor I should need or what should I do? Okay. Next thing, before going to the next section, we will search for some infinity values. Control F, INF, it is important, and no infinity values, but sometimes you will have infinity value. So it's better to check your data set for the missing or infinity INF values because in next step we will also check for the missing value. But if INF is written here, so the computer cannot distinguish between a real value and an INF value or INF keyword. So therefore, it will not be removed because in our code we will search for the missing value. And if there is INF written, it means the block is not missing. So it will not be clean that way. So save this data and the data name is merge descriptors and we will use this in part 2 for QSAR model generation and QSAR model training and all other things. Congratulations, you have started QSAR and you have learned how to generate or how to start the QSAR modeling and I have tried myself to give you a brief description of the QSAR. So I hope you learn something with me. If you are satisfied with my work and if you think I have not wasted 30 minutes of you, so please subscribe my channel, and you can also follow me on my WhatsApp channel for some news, for some updates, for some courses, and for some services. Thank you so much. See you in next video.